continue. So, looking at the table, you have here common saturated and unsaturated fatty acids. So, their category, number of double bonds, if there are any. And then we also have number of carbons. So, these are increasing. And their melting point. Okay, so looking at the number of carbons and the melting point, so we notice that for saturated fatty acids, the longer the chain, okay, so the greater the number of carbons, the higher the melting point. Okay, so meaning to say, you need more energy applied in order to melt the structure or melt the lipid. That is because of, so as mentioned earlier, so these are usually fats, no? So fats have higher component percentage of saturated fatty acids. Okay, so increased number of carbons, higher melting point. Meanwhile, for unsaturated fatty acids, so it is otherwise. Okay, so if we have greater number of carbons, and then we also have greater number of double bonds, so melting point goes slower. So it's easier to melt if you have higher number of carbons and greater number of double bonds. Okay, now that's for fatty acids. Another type of lipid would be your glycerides or glycerides, and they are either neutral or phosphoglycerides. So the distinction between the two is your structure. So when we say neutral glyceride, so these are glycerides where the glycerol or glycerol are attached with all fatty acids. So all of the substituents are fatty acid. But if you have phosphoglycerides, you have the presence of the phosphate group. So you have the substituent of phosphate, and then you usually have alcohol is attached to that phosphate group. And generally, glycerides are hydrophobic. So they are fatty acid esters of glycerol. And another term for glyceride is your acyl glycerol. Starting with neutral glycerides, so they are nonpolar, hydrophobic, they're not, meaning they dislike water. Uncharged esters form from glycerol and 1 to 3 fatty acid chain, meaning to say, for each of the carbon in this glycerol, so the substituent may be 1 fatty acid, 2 fatty acid, or 3 fatty acid chains. So it will depend. So the reaction between a glycerol and fatty acid, so if you only have one, so you will get a monoglyceride, and the byproduct is water. So if you have 2 fatty acids and then reacted with glycerol, so you have a diglyceride, and then the byproduct is water. And then if you have 3 fatty acids and then glycerol, so you have triglyceride, then you have, of course, water. Now, triglycerides make up more than 95% of lipids in the diet, and it is the main form of lipid found in the body. So recalling, we know that fats and oils belong to this category. So diba, we have fats and oils. Okay? And then, since glyceride is acylglycerol, so if you have 3, so that's why you have triacylglycerol. Now, there are different reactions in which triglyceride undergoes. So, one of them would be hydrogenation. So, as the name itself implies, you're adding hydrogen. So, if you add hydrogen, so you increase the number of hydrogens present in that fatty acid. Okay? So, here, it requires the presence of a metallic catalyst such as palladium or platinum. Okay, and this is the sample reaction. So you have oleic acid and then hydrogen exists as gas usually. So you have H2. So that is the stable form of hydrogen. And then you have here breaking of the double bond, forming single bond upon addition of the two hydrogens. So another example of this reaction, so if you have oil or fat and then you subject it to heat, so you will have the products glycerol and fatty acid. So they will separate. Okay? And then glycerol, upon subject to further heating and the presence of a catalyst, which is your potassium bisulfate, you will form aldehyde and water. Okay? So, from glycerol, your simplest alcohol, subject to further heating, you will form aldehyde and water. So, this is responsible for the odor of burnt food or burnt lipids. Okay, so that is acrolein. So acrolein therefore is an unsaturated aldehyde because it is an aldehyde with a double bond present. And of course, the other product or the byproduct is water, which evaporates. Diba? So, ma-evaporate ang water. Next reaction, so saponification. So, you have there soap making, or the principle behind soap making. So, this is a reaction, particularly a type of hydrolysis with the presence of a strong base. And it is the reverse of esterification. Okay, so if you have say an oil or fat, you have lipid source and then you react it to this strong base, so you actually form soap. Okay, so this is the principle or reaction, and soap is a product wherein you have a mixture of sodium or potassium salts, because the strong base may either be of sodium or potassium, depends. Okay, sodium hydroxide or potassium hydroxide. And you have fatty acids. Now, one example, if you have ethyl acetate, which is, so unsa ganin ni siya? What type of, so this is an ester, diba? And then you have a strong base. So for this case, you have sodium hydroxide. Then you subject it to heat. You form a salt. So you have here fatty acid. So sodium acetate or sodium ethanoate. And the other product is alcohol. So glycerol. Okay, that's why you have glycerol present in a lot of soap products. So glycerol is a simplest form of alcohol present in a lot of soap products. Now, how do we distinguish soap from detergent? The main distinction is your raw material. Okay, wherein soap is made from natural materials. You have fats, oils, while detergent is made from synthetic materials. In terms of formation, so the product of soap making, you have fatty acid, salt, and the strong base. Or what do you need rather is the strong base and the fatty acid from your fats or oils. And then for detergent, you have alkali or the strong base again, and sulfuric acid. So sulfuric acid is inorganic acid, diba? Now in terms of its effectiveness in hard water, so it is distinguishable by the formation of scum. So what is this scum? So it is actually sediments. Okay, and in terms of environmental impact, so soap can be easily broken down by bacteria, meanwhile detergent cannot. Okay, so what is scum? So when you have here hard water, there is formation of white na deposit. So this is because of the affinity of your soap. Because remember that you have there your fatty acids and they are hydrophobic. So they don't completely bind with water. Meanwhile, if you have soft water, meaning to say you have, say, purified. Example here, purified and then hard water. It could be tap water, or say from outdoors, outside. Hard water still contains minerals such as calcium and magnesium. Okay, and then they are in form of ions. So they interfere with the bonding of soap molecules. So the hydrophilic portion of the soap. Okay. So hence the question, if soap drops on the floor, is the floor cleaned, or is the soap dirty? So that's a trick question. No, usually it is in the humor. It's been ipaagi siya humor. If soap drops on the floor, is the floor cleaned or is the soap now dirty? Okay, so there's no definite answer. So it depends. Okay, so that's how we distinguish soap from detergent. Now, saponification. So there are two possibilities. If your base is a sodium, so you have sodium hydroxide, of course your soap is sodium salt. You have sodium salt. But if you have potassium hydroxide, you also have potassium salt. Okay, so the common one is there's always a presence of heat and your strong base. Okay, so heat and then the strong base. Of course, your fatty acid chain. Okay, so you form glycerol or alcohol. Then you have your soap. This is the salt. Sodium or potassium salts. Alright? Now, another reaction would be rancidity of triglycerides. Usually, this is observed in food. So, they develop unpleasant odors and taste because of hydrolysis. So, the reaction behind this is hydrolysis by bacteria. And then, of course, air oxidation. Okay, so that's why there are some food that require immediate consumption. Okay, and otherwise, you will have the rancid food. So, for example, we have butter. So, if it is exposed to air for a long time, and of course, bacteria will interfere, so you get rancid butter. Sometimes, if the temperature is high, it is favorable by bacteria, so you get rancid food. Okay, so far, we have covered triglycerides. And then, this time, let's proceed. So, triglycerides are said to be hydrolyzable. So, they can undergo hydrolysis. So, such as earlier. So, we have hydrolysis by strong base. So, saponification. And then you also have rancidity. Okay, and also hydrogenation. Okay, so for non-hydrolyzable, so these are some of the examples of the types of lipids that do not go the same mechanism as the previous ones. So let's proceed to phosphoglycerides. So earlier, triglycerides, they are neutral because all of the substituents are fatty acid chain units. But this time, for phosphoglycerides, also known as glycerophospholipids, you have the presence of your phosphate group and alcohol substituent. Okay, so they are similar to fats in terms of properties, and their main function, take note, is membrane components of cells throughout the body. So we have here in all glycerophospholipids lecithin, perhaps familiar, cephalins, and phosphatidylinositols. So these are always unsaturated when we speak of carbon 2 of the said glycerol. And then the third group is, so you have there the presence of phosphate group. So again, you still have your glycerol, the 3-carbon backbone of your structure, but this time phosphate and alcohol substituent is present in carbon number 3. Okay, it is always attached to carbon number 3. So one example of lecithin, it is said to be an important constituent of nerve and brain tissue. It is also known as phosphatidylcholine. And one specific source would be egg yolk. Okay, so this is the structure of lecithin, where you have here glycerol. Okay, and then the first fatty acid, you have saturated. Okay, for carbon number 2, you have here unsaturated. And for carbon number 3, you have the phosphate group and the amino alcohol. So, meaning to say there, choline is an amino alcohol. Okay, so this whole structure is lecithin. So, where do we find lecithin in food? So, it is said to be emulsifying agent, meaning to say, whenever you have two substances that do not mix well, such as oil and water, this emulsifying agent will be able to stabilize that mixture. So, emulsion is the dispersion of two liquids that do not normally mix. So, oil and water is an example of an emulsion, and emulsifying agent stabilizes that. So, for example, you have here in milk, since milk has different components aside from water, carbohydrates, lipid, protein, vitamins, minerals, it is a challenge to homogenize or to stabilize. Okay, so that's why we need emulsifying agent. So we have casein. Okay, so their main goal is to homogenize or to stabilize the mixture. Okay, so milk is an emulsion of butter, fat, in water. So you may not always see it, but the fresh milk from cow, for example, so over time, there will be separation of water and the butter fat component. That's why we need emulsifying agents. Okay, so that's for glycerides. So next we'll have non-glyceride lipids, where there is different examples or different specific types of non-glyceride lipids, and then we also have your complex lipids.